All right, hello everyone. My name is Michael Heuer. I've been working remotely for UC Berkeley from here in Minneapolis for going on 6 years now. I think the way things are going, I might be doing that for quite a while yet. So welcome to Minibar number 15. I'm going to start out the session today by talking about a workflow tool called Nextflow. And the question I'm asking is, can these workflows written in Nextflow be useful for more than science? Nextflow was developed for bioinformatics applications, the confluence of molecular biology and computer science. And the question I'd like to ask by presenting here to Minibar is, are these workflows and some of their properties useful for just general purpose types of things? So to give an overview of the talk today, I'm going to talk about a few properties of workflows. One that is that they can be repeatable, reproducible, and scalable, and I'll define those in a bit. Then I'm going to go through sort of a contrived example of a Nextflow workflow that, even though it's pretty simple in what it does, it attempts to address those concerns. And then finally, I will talk about a real-world workflow that's being used in production to analyze COVID-19 clinical samples. All right, so repeatable, reproducible, and scalable. The definitions I'm going to use here are as follows. For a workflow or a process to be repeatable, the question is, if you had to do it again on the same hardware and with the same data, could you do it? Is the process repeatable? The next level of reproducibility is whether or not this is reproducible. Even if you can do it the same on your own hardware and on the same data, can somebody else reproduce the results that you produced? And finally, is that process scalable? If it works well on a small set of data, will it scale up to handle a large set of data? And so in the scientific field, there's been a lot of work lately on making scientific results in terms of publications more reproducible, in that not only are methods for what you might be doing in the wet lab, in the chemistry, well documented, but that the software and the software analysis that you do for a paper have those be well defined and reproducible as well. So this paper that was published in 2013, the authors outlined 10 simple rules for making computational research reproducible. And the gist of this paper is that these things that you might want to do to make something reproducible are good habits, and not only that they make the work more worthy in the scientific community, but it's also helpful for yourself. And as they point out here, in other words, good habits of reproducibility may actually turn out to be a time saver in the long run. By having good habits, it helps you and it helps others. So outside of science, this lack of reproducibility can be a burden in all sorts of scenarios. So say a colleague leaves your group, and you suddenly notice the monthly log error report stops working. You guys start doing a little digging, you find out that it's a lot uglier than you thought. And I'm sure people have run into situations like this where something's running in somebody's home directory and It worked well enough to run, but is definitely not repeatable, reproducible, or scalable. And so for this talk, the example I'm going to use is along those lines. It's kind of a simple process, but one with a few steps, which makes it a workflow. So the process here would be to find all the log files in a set of application directories, gzip them to an archive directory for long-term storage. A second step would be to find all those log files, do the same find, grep for the serious errors, those containing lines in the log files containing the word error, and then gzip them to a separate errors directory. And then finally send out an error report via email, Twitter, Slack, whatever method of communication your group uses. So that leads us into Nextflow. What is it? So Nextflow is a programming language. It is a DSL. It provides some nice syntax for data flow and for processes. So you can set up data flow across a bunch of files, a bunch of steps in a process, and implement those steps as processes. Nextflow is pretty easy to get started. I can point you to the documentation at nextflow.io for more. And the example that I'm going to be running through in this talk is up on GitHub, and this is the repository. So we're going to take that simple set of find commands written in Bash, and we're going to re-implement those in the Nextflow language. And so this is the Nextflow file representing that workflow. And here at line 13, we're doing essentially the find. We're going to create a new channel, a new data flow channel, by looking in the log directory for files ending in .log. Then in the scripted analysis, we had two separate find statements. We can use just the one find here and tee the results into two separate channels. So that allows us to have two separate paths through our workflow. Then we've established a process, just a Nextflow syntax wrapper around the gzip command. And so here we're just going to gzip the files that come into this process as inputs and then publish them to a directory specified by the archive parameter. The next process definition we have here is just a wrapper around grep. Again, we're going to get the file from the input to our process. We're going to do the grep and then put the output into a new channel for further processing. Then we have another step, another process wrapper around gzip again. This time we're going to publish into the errors directory. And then finally a placeholder for the actual sending of an error report via email, Twitter, Slack, whatever your notification mechanism might be. All right, so to run this workflow, in this directory here, I have a clone of that GitHub repository. So you can just say Nextflow run in the name of your workflow, and Nextflow will execute that workflow. And so here we see the breakdown of the different processes that were run, the number of times they ran. And because we're running this on a local machine with the local Nextflow executor, these steps were parallelized using multi-threading on the single machine. And so we can see we have our gzip logs in archive and our error logs in the error directory. And so the first question is this workflow repeatable? And we can just clean things up and run it again, and we get the same answer. So yes, we've established that we now have a repeatable workflow. The next question is this workflow reproducible? Could somebody else use this workflow we wrote on their own hardware and with their own data? And so here I've just moved to a brand new directory on my machine. It has a log directory, a logs directory with a log file in it. Now I could go and clone that repository here and use it again as a working directory. But Nextflow has interaction with Git and other source code repositories built in. So what we can do is say Nextflow run and then give the name of a GitHub repository. Nextflow will pull the workflow from that Git repository and then run it here on our own data. We're specifying our own log directory to run. And so we get gzip logs in the archive folder and errors in the error directory. So have we got to reproducible? Well, this is a pretty good start. One possible problem is that my local computing environment may not be the same as the one that the script author used. Maybe I have a different version of gzip, or maybe I just don't have the dependencies necessary to run the workflow. So how do we make it more reproducible? The answer to that is that we use containers to do the execution in. And so support for Docker and Singularity is built into Nextflow. So all we have to do is run the same command, pulling the workflow from GitHub, but now we're specifying a Docker container to use in order to run all the analysis steps. So now I can pretty much, I feel comfortable saying that this workflow is now repeatable and reproducible. Anyone that has Nextflow and Docker or another container technology installed on their local hardware should be able to run this on their own data and achieve similar results. So the last question I want to talk about today is scalable. So if we have a workflow that works just on a small data set, how do you make it scale up to handle many? So the previous examples, we were just running on a single machine using Nextflow's local executor. And probably the biggest set of functionality that Nextflow provides are a set of different executors for all sorts of different environments. So here in the Nextflow documentation, we're describing the local executor. It's the one that's used by default. It just parallelizes your processes by using the threads you have on your CPU. Nextflow also provides support for lots of on-premise cluster computing environments. These will be run by a job manager such as Sun Grid Engine, LSF, Slurm, PBS, Condor. There are, this is the documentation for using the Slurm resource manager. This is one of the on-prem cluster management softwares. And in order to run our workflow on a cluster, all we have to do using Nextflow is set a process executor configuration parameter to Slurm in our config file. And then we would run the workflow just the same. It would check the workflow out from the Git repository. It would submit the jobs to Slurm. The Slurm jobs would run the jobs using Docker, we would have a repeatable, reproducible, and now scalable workflow. Nextflow also has support for cloud environments. So for example, you can, instead of using a cluster environment with lots of machines or virtual machines that need to be set up ahead of time, you could use something like AWS Batch, where the job is actually handled for you by the AWS service. And again, with Nextflow, in order to enable running on AWS Batch, all you have to do is set some configuration properties, starting with the executor, setting that to AWS Batch. The details with AWS and with some of the other cloud providers are a little bit more involved to get running, simply because you have to provide setup of your credentials and any sort of network and security parameters in which you want to run. So we have repeatable, we have replicatable, and we have scalable. So that's sort of all I have there for the simple example. Next, I wanted to move on and show a real world example of a workflow built on Nextflow. And the one I wanted to talk about today is called Viral Recon. This was developed at a virtual biohackathon that the bioinformatics community had in April of this year. There were a lot of people working from home, and we were itching for some collaboration. This virtual biohackathon was set up, and we had an amazing turnout. There were more than 500 people involved. And so there were a group of us that were interested in using Nextflow to help build analysis workflows for COVID-19 samples. And so at this biohackathon and after, it was a great international collaboration, people from all over the world. And we built this in the context of a community called NF Core. And that's sort of a large community of folks who are using Nextflow to do bioinformatics workflows. And so this is the GitHub page for Viral Recon. And I'm not going to go through the details of what the pipeline does, but suffice it to say, this has a lot more steps and a lot more pieces of external software that need to be gathered up and executed in terms of a workflow. So the basic analysis that's being done here is that in the clinic, somebody takes a sample of a patient and then goes through a sequencing process. This is more in-depth than sort of the regular COVID-19 PCR-based presence or absence test. This is an analysis, this is a sequencing of that sample to actually determine the sequence of the virus itself in that individual. And so a bunch of bioinformatics tools are used to process the sequences that come out of that sample. The end result is an assembled version of the virus genome and a comparison, a diff between the viral genome in that sample and the other viral genomes that have been observed. And so with those results, you can follow how different strains of the virus are migrating across populations. And so just like our simple example, to quick start the Viral Recon Nextflow workflow, you can just Nextflow run nfcore/viral recon, and it will pull what you need directly from GitHub. And then use a test profile, and then another configuration profile based on where you might be running. So for local, I would use test and Docker. The test profile just sets up a set of minimal data such that you can test the pipeline itself. And so we may run this locally on our machine using the local executor and Docker to run all of the different processes. And so this takes a while to churn. It needs to download a lot of data, and the Docker image itself is rather large. It had, I don't know, I think it's almost 25 different applications built into that one Docker image. But at the end of the day, you get an assembled genome of the COVID-19 virus that was found in that sample. And so I just wanted to share again links to some resources that I've presented here, to Nextflow itself at nextflow.io. The example workflow I showed is on my GitHub page at Minnebar15-workflow. The NF Core community hosts the viral Recon workflow and lots of other Nextflow workflows for doing bioinformatics. And the viral Recon workflow itself is at nfcore/viral Recon. And I'd like to thank everyone. And I'm not sure exactly how questions will go, but I look forward to any questions you might have. And thank you for your time. All right, I see at least two. Nope, I see one question on the chat. The question was from Jordan Peacock. What happens if you try to parallelize something that won't? For instance, if there's a file record lock. That is a good question. So the processes are parallelized by Nextflow in terms of how the processes send data to one another. So you have these input and output channels, and they're essentially queues. And so when a process receives input, it pulls them from the queue and it puts its output into a separate queue. And so if one individual process is sort of stuck or held up, there still will be things going into the output queue from other processes. So Nextflow is sort of able to work around the downstream. It will run jobs that are not necessarily downstream of the one that's stuck. And so the detail of how that works comes down to the execution environments that Nextflow is running on. So sorry, that was probably a long answer. And another question, is the slide deck available? Can I get the GitHub link again? Yes, I will put the GitHub link right here in the chat, and then I will also put a separate link for the slides, and I'll put the slides up on GitHub as well. All right, if there are no other questions, I'd like to thank everyone again for their time. It's been, I'd much rather be doing this in person, but I guess we got to do what we got to do. So thanks.